All right, guys, so we are going to go on into section 2 today, which is all about cell division. There'll be a couple notes that you'll need to take in red, and you'll be turning those in after we complete the next section of notes. So you can just add to your Chapter 8 notes from last week. I didn't highlight as many things as I usually do because we'll have another assignment later on in the week, and it'll go over all of the steps in detail. So I'm trying not to be too repetitive here. So cell division in prokaryotes. We'll start there because it's a little simpler than eukaryotes. Why is prokaryotic cell division different than eukaryotic cell division? Well, it's different because prokaryotes lack a nuclei and membrane-bound organelles. Prokaryotes also have a single chromosome that is circular, and it's not coiled around any proteins, which we call histones. So here you can see the prokaryotic DNA. It just hangs out inside of the cell. Eukaryotic DNA is located in the nucleus, and it's tightly coiled up. Kind of looks like wow, I'm going to date myself here, but if you guys ever used, an old phone that has a cord attached to it that's not a cell phone, it kind of looks like that. Or maybe you could even think of it like a slinky. You played with those whenever you were a kid. So we call this division binary fission, and it's when a cell is going to basically split right in half, and they're going to be identical to the parent cell. So here's my cell. You can see all these little yellowish orange dots are going to be ribosomes. It has a plasmid in there, and then it's DNA. So the DNA is separate from the plasmid. And if you take genetics whenever you're a senior or even a junior, a plasmid actually allows a cell, these are only bacteria cells, to gain some type of an advantage overall. So this could be an advantage such as bacterial or antibiotic resistance, or the ability to mutate, or the ability to combine with other types of bacteria so that you end up with like a super bacteria. These bacteria have some crazy plasmids, but I'm not going to get too much into those, at least not in biology. So steps to binary fission. First, the DNA is copied, which is going to leave 2 identical chromosomes attached to the inside of the membrane. So here's our OG DNA, it's black, and the copied version is going to be blue. The plasmid also copies. A new cell membrane is going to begin to develop between the 2 DNA copies, and the cell is going to grow until it reaches twice the original size, and then it's just going to pinch inward. So the original cell isn't actually dying, it's just splitting in half. So a new cell wall is going to form around the membrane, and the prokaryote is going to split into 2 identical cells. So here we have our original cell, here we have our new daughter cell. Now in eukaryotes, this division is going to occur in both the cytoplasm and the nucleus. There are 2 different types of cell division in eukaryotes, and that's dependent on if the cell is an autosome or a gamete. A mitosis is going to be forming from cell division in eukaryotes. It results in new cells with genetic material that is identical to that of the original cell. It occurs in organisms during growth, development, repair, or asexual reproduction. Asexual reproduction is the production of offspring from one parent. So it looks basically like this. You have your original cell up here at the top. It copies its DNA. You can see it starting to divide. Here we have 2 new cells, and now they are separate entities. Meiosis is cell division that occurs during the formation of gametes, the sex cells that are haploid. It reduces the number of chromosomes by half in the newly formed cells. These haploid sex cells have the potential to join with other haploid sex cells to form a diploid cell, and that is how you begin. So sperm and egg are going to come together. They're both haploid, so on their own they cannot form a baby, but whenever they combine, now this one cell is going to have a full set of chromosomes, and that is going to allow it to develop into a baby. It's very important that they are haploid, because if you had a cell that was diploid trying to merge with another diploid cell, well, you would be double the amount of chromosomes, and you wouldn't actually be able to form a viable baby. This is the cell cycle, and eventually we'll be drawing this and we'll be coloring it, and I'll go in depth with each part. So the cell cycle is a repeating set of events in the life of a cell that includes cell division and the time in between cell divisions. Interphase is the time between cell divisions. It's divided into 3 different phases, the G1, the S, and the G2. And you can see that the cell spends the majority of its time in these 3 phases before cell division. Just this tiny little chunk is cell division. Everything else is interphase. The cytokinesis is the time immediately after mitosis, and that's going to divide the cell's cytoplasm into 2 new cells. Now, even though cytokinesis isn't directly a part of the cell cycle or mitosis, we kind of lump it into mitosis. So it's right off here at the end. We just kind of clump it together with mitosis, even though it's not actually a real portion of it. But it is part of the cell dividing, so that's why it's in there. The cell spends most of its time in these 3 phases of interphase. G1 phase, which is the gap 1 phase. This is where the offspring cell from the previous division is going to grow to a mature size. So G1 is for growth. The S phase is the synthesis phase. This is where the cell's DNA is going to be copied. A fancy word for copy is synthesized. G2 phase, or the gap 2 phase, this is right after the S phase happens, and the cell is going to grow again, and G2 acts as a checkpoint to make sure that everything is ready for the cell to go into cell division. At this point, a cell can also leave after G1, and instead of moving into the S phase, it's going to get off at called the GO phase. In GO, the cells do not copy their DNA. They don't prepare for cell division because they are not going to divide. Fully developed nerve cells stop dividing once they're mature. So they spend the rest of their life in the GO phase, which is why you don't get new nerve cells. If you ever get a really deep cut, my mom cut her finger, probably about 25 years ago. And she cut it so deep, she was making dinner, that she never actually regained feeling in that part of her thumb because she cut through a nerve. That's what a nerve cell looks like. Mitosis is divided into 4 stages, and they occur in order to distribute the cell's copied DNA into the offspring cells. So I did also say that a cell could stop at the G2 phase. So if something isn't right with the DNA after it's been copied, it's going to be checked over in the G2. And if there is some type of mutation and your cell catches it, it's going to be like, hey, we don't want to have this portion replicated. We don't want to make more cells with this mutation. So it'll actually tell that cell to go through a programmed death or apoptosis. Getting back to mitosis, though. The 4 stages are prophase, metaphase, anaphase, and telophase. You may have heard these abbreviated as PMAT. The first stage is prophase, and this is what a cell looks like in prophase. The DNA is going to shorten and it's tightly coiled into chromosomes. The chromosomes have already been copied during the S phase of interphase, and these can be seen under a light microscope. The nucleus and nuclear membrane are going to break down and disappear so that the chromosomes can move throughout the cell, and you can see that there's no big black balls anymore. It is kind of separated. You can see the individual strands. It's a little easier to see whenever we draw pictures of representations of what's going on. 2 pairs of dark spots called centrosomes appear next to the disappearing nucleus. Each centrosome in animal cells contains a pair of small cylindrical bodies called centrioles, and plant cells, they don't have these centrioles. The centrosomes are going to move to opposite poles of the cell during prophase. As the centrosomes separate to the opposite ends of the cell, spindle fibers made of microtubules come out of the centrosomes in preparation for metaphase. The many spindle fibers are grouped together in a structure called a mitotic spindle. The mitotic spindle is going to divide those chromosomes equally. So here you can see these centrosomes, these lines coming off of the spindle fibers, and they are connecting to the DNA that you have down the middle here. If you wanted to look at an actual cell doing it, that's what it looks like. The second phase is metaphase, and you always think of metaphase as meeting in the middle. During this phase, the chromosomes are the easiest to identify, and this is where you would make a karyotype if you wanted to make a karyotype. The mitotic spindle is going to move the chromosomes to the center of the dividing cell, so it will look just like that. This picture here is a plant cell. Down here we have an animal cell, and you can actually see the spindle fibers in the animal cell pushing those chromosomes. This one's just a little easier to see. They're colored in two different colors because one is going to represent the original DNA and one is going to be the copied DNA. Anaphase. The chromatids of each chromosome separate from the centromere and slowly move toward opposite poles of the dividing cell. After the chromatids separate, they are considered individual chromosomes. So there you can see them being pulled apart. They're no longer right in the middle. Those red portions are actually the DNA. Here is the chromosomes being ripped right down the middle. Telophase. Final stage of mitosis. The chromosomes reach opposite ends of the cell. The spindle fibers disassemble. The chromosome is going to return to a less tightly coiled form of chromatin, and the nuclear envelope forms around each set of chromosomes. The nucleolus is going to form in each of the new cells. So here you can see there's starting to be a division here. You have two completely new cells separating. There it is. They're kind of still formed in the middle, though. We got to cut away the cell membrane. During telophase, a process called cytokinesis begins to divide the cytoplasm between the two new cells. In animal cells, the cell membrane is going to pinch inward in the middle of the cells, and this area is going to be called a cleavage furrow, and that is what it looks like in an animal cell. Cytokinesis in plant cells is a little bit different because they have a cell wall. So a vesicle from the Golgi apparatus is going to join together in the midline of the dividing cell to form a cell plate. A cell wall is going to form from that cell plate, and it's going to continue to divide the new cells. Just like in an assembly line, the cell goes through 3 checkpoints to make sure it's ready to divide. The G1 checkpoint is going to check over, see if the cell is a suitable size, and make sure that everything is needed for cell division is there. Then it's going to send it into the S phase if everything is good to go. If not, it's going to kick that cell out at the GO phase. DNA synthesis, that is going to actually have a checkpoint at the G2 phase. So DNA is going to be repaired. It's going to check the results of the replication. If something is missing, then it will, again, kick that cell out. And then there is a mitosis checkpoint. If a cell passes this checkpoint, the protein signal that the cell should go on ahead and divide. Otherwise, it's going to make it go back to G1 again. The proteins that regulate cell growth and division are coded for by genes. If a mutation occurs on one of these genes, the proteins may not function properly and get through a checkpoint that it has not been cleared to pass. So it's kind of like Monopoly. You'll either collect your $200 and continue on, or you'll end up going to jail. When a disruption occurs, it can result in uncontrolled cell growth, which is how a tumor forms, and you end up with cancer. Cancer cells do not respond normally to the body's control mechanisms, and it leads to increased cell division. So basically, these cancer cells bypass all the checkpoints. They don't even take into account that there is a checkpoint, and they just divide and divide and divide. And as they're doing this, because there's no checkpoint, that DNA just keeps mutating and mutating and mutating. And that is how a tumor grows and gets bigger and bigger. So there we have a little tumor cell attached to some type of an organ. The next section is on meiosis, but we'll stop there for now. Now you may have noticed that there wasn't anything red in my video. That's because I used the PowerPoint that I use in class. But on your PowerPoint, if you click on the slides, there is some red portions in there. So use the slides for your notes. And it's actually done right here. There it goes. There, so here you can see the red portions. Not everything is in red, but there are some red slides. Sporadic. They're out there. All right, guys. I will talk to you in a bit.